Hello and welcome today to today's webinar about the online platform SFARI Viewer. I'm Rachel Jerd, editor of SFARI.org, and our speakers today are Alistair Ward and Chase Miller. Alistair and Chase co-founded Frameshift Genomics, which is the company that developed SFARI Viewer in collaboration with SFARI's informatics team. Natalia Folowski, Director of Data and Analytics at the Simons Foundation, is also joining us on the presentation, and she'll be here to answer any questions relating to SFARI Base. SFARI Base, as you'll hear during the presentation, is the critical first step for requesting access to all SFARI data, including the data sets that are part of SFARI Viewer. I also wanted to mention that SFARI Viewer is one of a number of different web-based tools that are available to visualize and analyze SFARI data and other related data that's relevant for autism researchers. So I've listed a few here, and more information can be found on the website at the bottom of this slide. So before we begin the presentation, one final housekeeping point that I wanted to mention is that we will be taking questions from the audience at the end, but you don't need to wait till the end to submit your questions. So please ask them at any time during the presentation by submitting them via a chat window on the side of your screen, and we will relay those to the presenters. And if we don't get to all of the questions, we will definitely respond back to you afterwards. Okay, so now on to Alistair and Chase. Thanks, guys.
Thank you very much for the introduction, and thank you very much for the SFARI team for giving us the opportunity here to spend a little bit of time explaining this new tool that we've released in collaboration with Frameshift and SFARI. And this is called SFARI Viewer. And so we're going to spend the next half an hour, maybe a little bit longer, explaining what this tool is going to do. So I wanted to thank everybody who's joining us on the call today for taking some time out to learn a little bit more about what we've been working on and hopefully how this may be useful to aid in your own research or use of SFARI data. So the primary goal of the presentation today is to walk through a few questions that will help maybe pique people's interest in what this tool can do and maybe inspire them to afterwards to go take a look at the tool and some of the other SFARI tools that are available for accessing the data they have. So we're going to try and make sure that by the end of this, people will be able to answer these 5 questions. Essentially, what is the SFARI Viewer and why we developed this and what the role we're trying to fill is. Secondly, we want to describe who this tool is designed to be used for, and then describe in a little bit of detail how you will be able to get access yourself to be able to use this tool in your own work. We're going to mention some of the resources that are available that will help people learn a little bit more about what the tool can do and how they can provide feedback, comments, and maybe engage with both the team here at Frameshift and at SFARI on how they can use this tool themselves. And the bulk of the presentation is essentially going to be doing a live demonstration of the tool itself and walking you through at a very high level some of the features that are available. This isn't going to be a very in-depth, detailed view of everything to show off all of the functionality, more just a broad look at the general things that the tool can do, and hopefully then you can investigate or work with us to look in a little bit more detail. Okay, so SFARI Viewer, this is a new tool that we've developed, as I say, in collaboration with SFARI. And the SFARI Foundation has generated a huge amount of really rich data to support the autism research community. And a lot of this is in data files that are difficult to access to a lot of people and don't necessarily know where those files exist. So what we've developed is a web-based platform that is intended to try to make it easy to navigate between the user and those files, and then display all of the information that we have available to the user in as straightforward and intuitive a manner as we can. So really we want to have data visualization be front and center so that everybody can really get access to this data and be able to explore that data and understand not just what data exists, but how that data relates to each other, and hopefully allow them to generate their own insights on the data or come up with hypotheses that they may want to test with additional methods. Finally, since this tool is providing an interface between the user and some of the genomic data files, this tool also acts as an access point to allow additional web-based data analysis tools to use that data to allow some more focused analysis. And we'll show some of that as we walk through the demonstration today. So we've made a great deal of effort to really focus on making sure that these tools are really driven by visual means. And the goal here is to make sure that everybody who is authorized to see the data can make use of this tool to search this very rich data set. So this would mean that it's not tailored to specifically work with people with a bioinformatics or a computational background. We're really hoping that people that come from all sorts of backgrounds, whether that be biological, whether they come from a clinical background, as well as a sort of a data science background, including PIs, their postdocs, graduate students, essentially anyone in those teams to be able to work with this data. There is obviously one big caveat in that this data isn't available publicly, so only individuals that have been authorized to see the data will be allowed to use this tool. And so that sort of moves us to how one would get access to all of this data. And as mentioned, authorization is mediated through SFARI base. So you would need to go to the address on screen here and request access to the data. So in this scenario, the PI will be able to go into SFARI base and make a request to view all of the data necessary for SFARI Viewer to run. And so I would recommend in making a request that you specifically mention that you're trying to gain access to SFARI Viewer to ensure you get the access to the correct data. And also in that request, you should indicate which team members you also want to be able to access the data. They will need authorization themselves as well. And so authorization is then mediated through a tool called Globus. You will need to get a Globus ID, and the SFARI team will authorize your Globus ID to access the data. And once that is complete, then you will be able to use SFARI Viewer and use all the functionality that I'm going to show in a short while. I will make just a couple of points here. There are going to be investigators who have already got a Globus ID, have already been through SFARI base to get authorized for different projects. So just because you have an authorized Globus ID, that doesn't necessarily mean that you have access to the data necessary for SFARI Viewer. So if you use your Globus ID to get into SFARI Viewer and it appears blank, that would basically mean that you don't have access to the correct data sets. So we'll need to put in an independent request through SFARI base to gain access. And we have noticed a couple of times that some people have struggled a little bit in this process. So if in going through this you find any problems at all, please don't hesitate to contact either the Frameshift team or the SFARI base team. Either way, both SFARI base and Frameshift will work closely with you to make sure that we can get everyone who gets authorized, get them access to the tool. So there are a number of resources that we've made available to try to make sure that the SFARI Viewer and associated tools from there are as easy to use as possible. And we really do want to try to engage the community and get suggestions or comments on the tool and features that they might like for specific use cases that they work with. So within the tool, there is an option to view a whole range of tutorials, and the tutorials are there to try to explain what each of the tools do, what each of the views within SFARI Viewer do, and try to give some examples of how this tool can be used to try to make sure that people can independently learn how to use the tool. There is an email support form within SFARI Viewer where we would encourage people to report any bugs or feature requests that they would like to see, or provide any general feedback to us. There is also a live online chat. So generally, if you're working with the viewer and have questions about the viewer, you can go into the live online chat, and so long as this is within sort of East Coast work hours, we will endeavor to ensure that we try to answer as quickly as possible and work with you in real time to resolve any questions that you may have. And finally, since the demonstration today isn't going to get down into all of the details, if any individual groups are interested in learning more, then we would be more than happy to set up some individual meetings with those labs where we can go into a little bit more detail about how the tool works and maybe discuss particular use cases that they may be interested in pursuing. So with that, we're going to jump into a demonstration of SFARI Viewer. So this is always a dangerous game to actually do a live demonstration, but that's what we're going to do. And we're going to use this on a couple of example data sets that we've generated that look broadly like the kind of data that is available through SFARI Viewer. So these example data sets will give us a pretty good set of data to show you how these tools work. So I'm now just going to jump over to the SFARI Viewer tool. So if you gain access to SFARI Viewer, once you get authorization and log in, then you will come to a screen that looks very much like this. And on this first screen, we have a description of all of the data projects that are available within SFARI Viewer. And so we've split the projects up into a few different groups, essentially grouping together all of the data generated through the exome sequencing project into a single project, and sequencing are separated out into separate projects. We've also ensured that data that has been aligned to different reference genomes are contained in different projects to make sure that people don't try to do analysis on data that exists in different coordinate frames. So the cards at the top show the projects that I've used most recently. The table below shows all of the projects that are available. But in this example data set, there are only 2 projects for us to play with, one based on exome data and one based on whole genome data. So if I click on one of the projects, that jumps into a dashboard for this project that gives us some very high-level details about the project itself. And down the left-hand side, we have a number of different options that are available for digging into the data. And the most interesting of these, and where we're going to start here, is on the analytics page. And so the analytics page is essentially a collection of individual charts that show how all of the different attributes associated with this project are distributed across all of the samples. So this example project has of the order 9,000 samples attached to the project, and then there's a lot of different attributes for which each sample has a value. So one of the first things that this page helps you do is just get a quick sense of all of the data that is available for the project. So by clicking on the Select Charts button, I get a modal that shows me all of the charts that I currently have selected and are viewable within analytics. And then I have a table that includes all of the different scores and attributes and quality control attributes that are available. I can search for anything that I'm interested in. So if I'm interested in, for example, looking at some genomic information about how many reads are in each sample have, how many of those reads are mapped, I can find all of the different attributes that are available and then choose which charts that I want to view. And so as an example here, this top chart is showing me a quality control metric associated with the genomic sequencing of this exome project. And so one attribute of interest is what is the median coverage of each sample. And so we can see here that the median coverage across this entire project is distributed with a median here around 50. So most of the samples in this project have a depth of around 50x. And we see a nice tail off to the right where we have some samples that even get up to around 300x coverage. Unfortunately, not too much down in this left tail here. So another very useful feature of this tool is it allows us to filter out samples based on attributes of interest. And that'll allow us to look at what the distribution of different samples are. So as an example, I can come down here and interact with these charts to select only the samples that have this communication score less than 60. And you'll have noticed as soon as I select this region of data, all of the distributions here had an additional chart drop out of the bottom. And so these new charts are showing me the distribution of each metric, but only for the set of samples that I just selected. And you can see immediately that there's far less samples in the filtered set than there are in the whole set, as you would expect. So if we want to actually compare what the distribution of the filtered set looks like compared with the whole set, we really want to remove the fact that we're constraining the y-axis to have the same scale. And so we have controls here at the top of the screen that allow me to relax some of these constraints and essentially let me look at what this distribution looks like for the sample set I've just selected. And so now you can see quite clearly that if I look, for example, at these IQ attribute distributions, that having selected the individuals that have a low communication score, that seems to give me samples that also score on the lower end of the two IQ spectrums that we have here. And one thing that's kind of fun, in my opinion, to try and play with this data is to move around these selections and watch how the different selections of samples will affect how these distributions look. So I can grab this section of that I've filtered and move it to select a different set of samples and watch how all of the distributions are affected by my change in sample choice. So as I slide this selection over to the right, selecting individuals with higher and higher scores on the communication metric, I can watch the distribution of these IQ metrics move in concert. So this gives me a suggestion that there is a correlation between these different scores. And so given that I can choose any different metrics and play with them in any way like this, this lets me just look visually to find out if there are connections between different sets of data and maybe come up with some hypotheses that would lead me to want to have a more in-depth study to check these for statistical significance. Another very useful benefit of this is that I can choose custom cohorts that I would like to look at. So as an example, I may want to go into a study that I've just seen and replicate the study that they just did. So I can come into here and take all of the conditions that they applied in that study to generate the samples that they had. So if I knew a study had been focused on children that had an IQ in the range of less than 90, for example, then I can select on multiple different charts to generate this cohort of samples that I want, and then use that set of sample IDs to then make sure that's the samples that I would use if I want to replicate the study. So if I was to do that, what I can then do is I'll go back to my original selection of a communication score below 60, and I'm going to jump over to the samples tab over here on the left of the screen. And this will now give me all of the samples that I just selected from the analytics screen. So if I was attempting to replicate a study and needed to know which samples I had, this is all of the samples that conform to the filters that I just applied. And so there's a lot of interesting information that is in this table. Also, in particular, I'll point out one of the things that bringing data visualization in helps us achieve is I can pull in distributions for each individual sample now and allow the user to compare across a whole group of samples. So where on the analytics page, I was able to look at how the median read coverage was distributed across the entire cohort, I can now see how that median read coverage distribution looks for each individual sample. So it may be the case that the median read coverage is good for every single sample, but they may have different distributions. One sample may, for some reason, have a bimodal distribution instead of a Poisson distribution. And being able to look at them all together and scroll down multiple samples and look how these different distributions compare allows us to quickly spot where there may be problems in data. And I also have a lot more information, like the pedigree of this particular individual. And so in this case, this would be this particular sample at the top is the daughter in a pedigree of 4 and is also the affected child in this family. I also have a lot of other information, like the variant count here, but this is also customizable. We want to ensure that the user can look at the data which they're interested in. So I have the option to jump in here and also include in this table any other scores that I'm interested in. So for example, if I was interested in the irritability score, I can include that in the table. And now this is something I can use to sort on. So I've limited myself to only the probands that have a communication score less than 60, and then within that group of individuals, I now have them ranked by their ABC irritability score. And so there's a lot of power to really manipulate the data that we're looking at and find the cohorts that we're interested in. Now, there's a lot more information available within this app. I'm only going to show one more view so as to not overwhelm people with too much detail, but I can now jump down if I'm interested in this particular sample, for example. I can just jump down to a sample page and view more detailed information about this particular individual. So I get a much more high resolution view of what the read coverage distribution looks like. I can see the relative distribution of SNPs and indels in the variance code for this individual and also their ancestry. And so all of this is then available for all samples, and I can search across all of this for every individual that we have. And again, so there's a lot more features available within this tool that allow us to dig around and understand more about the families, the individuals, and cohorts at large. But that's all we're going to talk about for now about this particular tool, other than the functionality that we have down here that allows a user to jump out to additional web-based tools to do more focused analysis. So if I click on here, this gives me the option of a variety of different tools that I can use to do some more focused analysis. So for example, if I was interested in this proband and the family, and I wanted to look at their genes to see what genetic variants were present in this family, then I can use this gene.iobio tool. I collect the tool, it's automatically showed me the family that we're looking at. So I'm going to be looking at this male proband and looking at their parents. And so in order to launch out to the tool, I need to provide a gene. So I'm going to type in, for example, SHANK3, and then as I click launch, this will launch out the tool that we're going to look at. So this tool can take, it takes a couple of seconds to load in. So I actually ran this a little bit earlier just to make it quick. What this tool essentially shows is a whole host of information around this one individual family. And again, there's a lot of information available in this tool. I'm going to try and stick at a high level. Again, if people are interested in learning more about the details of this tool, then please get in contact with us and we can really walk you through all of this. But essentially what you're looking at is in this particular gene that I've selected, I can see a track for the proband and each of the parents. And I'm looking at a particular gene model, and I can see what the sequencing coverage is across this entire gene. And then I can see on the top here, these are the variants that are present in the proband in this gene and the variants that are present in the parents. Now, what happens? It takes a couple of seconds for each gene to load because this is now connected securely to the raw underlying data in the project that you launched from. So all of these variants have actually been annotated in real time. So all of these variants show a color, and we have a little table here that summarizes all of the variants. And so it's telling us any of these variants, if they're present in ClinVar, and what their pathogenicity is within ClinVar, what their functional impact is, or the allele frequencies in known databases like Nomad, Exact, for example. And so for every gene we look at here, we get all of the variants, we get them all annotated, and we didn't need to know anything about how to get access to the data or the underlying resources to do these annotations. And what I did ahead of time is I actually provided 140 genes to this tool, and it still only takes it a minute or two to work through 140 different genes. And these are genes that have been generated as part of the SPARC project that have been curated to have a strong and reproducible known effect in autism. And so I may choose to look for this particular family. Do they have any interesting variants in these 140 autism-related genes? And so I can see that the tool has gone through and annotated the variants in all of these. And where we see an additional little glyph, this indicates that we've found a variant that we think might be interesting. And interesting is basically defined as being rare in the population. So, allele frequency of less than 5% in Nomad or Exact, that has a significant functional impact. So maybe there'd be frame shift or nonsense mutations, or unknown to be pathogenic in ClinVar. And so once the annotation is complete, I see a list of all of the variants that were found in this proband that were deemed to be interesting. And I can see these are all then stratified by the variant, the inheritance mode. So we have a single de novo mutation found here. We have some compound heterozygotes that were found across these genes, and also an X-linked variant in the FMR1 gene. So this is a tool that's actually available for free at gene.io, but there is an actual feature in here that is unique to being launched from SFARI Viewer, and that is we have this SFARI track that appears in the top of the screen. And what this allows me to do is select the SFARI track, and it will go and look in the SFARI data for any variants that are present in this particular gene, and I'm filtering these only by high or moderate impact variants in that data set. So I could be looking at a family of my own here, but then looking to see if there are any variants that are shared between the SFARI dataset and my own dataset. And in this case, there is nothing shared here. But I can click on any of the variants that my proband has, gain some additional information. So this is known as a benign variant in ClinVar. So I can click on this and find out what is known in ClinVar about this variant. And I also have a whole host of other details about the variant, and links out to additional tools that may be useful in looking at evidence of pathogenicity in this variant. So this is a really cool tool that has a great deal of functionality for really looking at an individual family level. But we may decide that we want to look not at one family, but lots of genes, but instead look at one gene across a different cohort. So in collaboration with Gabor Marth's lab at the University of Utah, we've also developed a tool that will allow us to look for enrichment in an individual gene based on a cohort that we're interested in. So for example, here I'm just jumping out to an example whole genome project. And I have again selected the probands that have a communication score less than 60. And my question here would be whether in any particular gene, if the variants that are present in this filtered set of samples is enriched as compared with the entire proband population within this particular project. So to answer that question, we have a tool called Cohort Gene that's also launchable directly from SFARI Viewer. Again, I need to provide this with a gene. So I maybe go take a look in the FMR1 gene. And when I launch out to Cohort, Again, what this is doing is fetching all of the data from the raw variant call file at wherever your project has stored it. So again, the user doesn't need to know where any of this data sits. This is securely passing on the data. It's going to do two things as this data comes in. One, it's calculating enrichment for variants in the cohort that I selected against the whole probands, and it's also annotating all the variants, just like Gene did, against all of these known resources. Since this is doing a little bit more of a calculation, it can take it a few seconds, and so in the interest of time, I also have it open here. So what we can see is in this FMR1 gene, these are all of the variants that were present in the samples from our example project. And I can filter on all of these variants based on a whole variety of tools. So let's say I'm only interested in variants that are less than 5% allele frequency in Nomad. I can select that frequency filter, reduce the set of variants down here. And on the y-axis in this scale is the enrichment of this variant in the specific cohort of data that I've selected. So I can play around and look at the variants here, and it looks like in this example set that there really is no enrichment of note. But the other thing that is interesting here, both in gene.ibio and in cohort gene, we're able to add our own files. So this is now just looking at the data that was launched out of the SFARI viewer from the example project, but I can come into here and add my own tracts. So I'm going to add my own sample here to compare against all of the data. So I'm going to choose user sample 1, and I'm going to take a link. So you can either select files directly from that you have stored on your hard drive, or provide a link to, for example, an S3 bucket on Amazon. So I'm just going to pick this VCF file that contains the genetic variants, and then I'm prompted to choose the sample. So there's multiple samples in this particular VCF, so I'm going to pull out the standard NA12878 sample, and I could, if I chose, also add the URL to the sequencing data itself to look at the coverage in the gene. And I can add any number of samples that I want, and every time I add, I just generate a new card here, and it's going to go out now to that file, pull all the variants from this gene, annotate them against all of the annotations we have down the right. And again, I can now look to see if this is a variant shared with the SFARI project, or in this case, the example project that we launched out. And so we can see the only variant in this particular case is not present in SFARI. So this is, again, a tool that lets us dig into the information in a single gene, look for statistical enrichment within a cohort that we select. So I'm always able to go back to SFARI Viewer, pick a new set of samples, and then look for enrichment in there too. So I think that's sufficient information on these tools for today. So I just want to thank everybody for taking the time to listen to the webinar. Hopefully this has made people aware of some of the tools that are available and maybe would like to go take a look at. So if you would like to learn more about how to gain access to the tool, then SFARI Base is where you need to go to look at how to gain access. The URL to SFARI Viewer itself is on here too. Again, in order to actually gain access, you need to have authorization first. And then for any feedback on the tool or how you would like to use it, please feel free to get in contact either with the SFARI team or with the team at Frameshift at the email addresses here. So I think that's a good place to finish the discussion here. And so it would be a good time then to move on if anyone has any questions.
Okay, thanks very much, Alistair. And thank you to all of you for being on the line. And if you do have any questions, please send those in to us on the chat via the chat box. And in the meantime, while we're waiting for questions from the audience, I'll start by asking one. So Alistair, I think you showed us 2 of these apps that are available through SFARI Viewer. Are there any other apps that that are available?
Yes, there are. So we have the whole tool is built on top of an API, a programming interface that allows us very easily to connect different tools in. So we already have a number of different web applications that are available. So the tools that we showed allow analysis of an individual gene and really digging into individual variants within a gene or within a set of genes, and also the cohort gene for enrichment. But we also have a couple of tools, one that allows us to dig into an individual sample and really understand the sequencing coverage information and other statistics associated with whether or not the sequencing experiment itself was good. And again, this is it's a real-time application that connects directly to the data files and generates distributions and information in real time as you watch, and gives you a good idea of whether the data for a single sample is good. We also have a tool that's called Multibam that allows us to look at these same sequencing quality metrics, but across a whole cohort. So then again, the philosophy here is if we can display as much information about a cohort at the same time, then we really get the human eye to be able to do what it does best and spot some of these errors where, you know, quite often we put some thresholds on data that things have to fall less than a particular number, but that doesn't often capture the sort of the nuances of the data that we have. So, so we have a couple of these tools that really let you dig into that data and let you make an assessment based on your own visual abilities.
Okay, thanks. And is it possible to share visualizations with colleagues?
Yes, it is. So to share any visualization or any page, all you have to do, it's really straightforward. You just copy the link and send the link over to your colleague. And as long as they have access and they're already logged in, the link will pull up as shown. So this is an easy way for two people to collaborate back and forth.
Thank you. Another question is, can you use your own tools from SFARI Viewer?
Yes, that is possible. So as I mentioned before, we built this really with the view that the tools that we supply are not going to cover every single possible question that anyone might have. So we've worked with a group before to ensure that they have their own tool and they have access. They already have access to SFARI data. So we were able to build a secure link to one of their tools. So they're able to use the analytics functions within SFARI Viewer to identify cohorts that they were interested in, and then use that to power their own R algorithms locally. So yeah, so this is something that we would encourage anyone who has tools that they would like to use to get in contact with us, and we can work with them to make that accessible. You know, obviously we have to do this to make sure that we preserve all the security and access control.
Okay. And what about additional SFARI data? Is there plans to add more data sets or at least additional samples, say from Spark, where participants are being added every day? Do you have plans to have that data incorporated in the near future?
Yes, so we're actually actively working to make sure that, so the Spark data set, for example, is growing, and we're working directly with the SFARI informatics team to ensure that this data continues to get updated. And so SFARI Viewer will continue to grow in what it has as the data at SFARI grows. So yes, absolutely. We hope that this tool is going to continue to dynamically grow as everything else grows, and continue to be useful across all the data that's available.
Okay, any questions from the audience that you'd like to ask? So if there are any questions that you think of afterwards or after you begin doing your own analyses, you can always reach out to SFARI or Frameshift via email or any of the other ways shown on your screen there. And I also wanted to mention that if you wanted to view the webinar again, or have colleagues who you think might be interested in viewing it, a recording is going to be made available on our website, safari.org, probably within the next few days, and we'll also include it in the next SFARI newsletter that goes out, and it will be posted on social media. So I want to thank our speakers today, Alistair and Chase, for going through this platform. It definitely helps, I think, to make it more intuitive for users. So thank you. And I want to thank all of you for being on the line. So that concludes our webinar for today.